Good morning. Well, we're in the home stretch. Chapter 14, final chapter for biology for majors, biology 1302. This chapter is on speciation and extinction. Speciation is the process where new species evolve from older species, and extinction, we all know what that is. That's when a organism completely disappears from the life on the planet. So, what is a species? There are lots of different definitions of what a species is. There's some that take certain aspects into consideration while others don't consider those. But in short term, in just very quick language, a species are distinct groups of organisms that interact with each other and reproduce, producing viable offspring. In evolution, new species will form while others become extinct. Again, we will talk in well, not great, but in a little more detail here in a few minutes about the way speciation happens and the characteristics of what has to happen for new species to form. So, smaller evolutionary changes will accumulate in a population, which is called microevolution. Microevolution is those small mutations, those changes in DNA that don't affect the overall size, scope, or other characteristics of an organism. The human being is micro-evolving at all times. We share, a great deal of our DNA. Some of that DNA between individuals is different, and as that DNA mutates, it could evolve. Eventually, not in all cases, but eventually it can lead to what is called macroevolution, which is much slower and results from large-scale changes. An example would be in the birds. There are birds that are swimmers, there are birds that are perchers, there are birds that are climbers, there are birds that don't fly. That change from being a bird that flies to being like an ostrich, a bird that does not fly, that kind of change does not happen as a result of microevolution. That is the result of what is called macroevolution. Microevolution can change very quickly. Bacteria, for instance, can reproduce or can mutate and evolve very quickly, making those small changes. But these large changes that result in speciation have to happen over multitudes of generations. They don't happen just, within a few years or decades. These things take hundreds, thousands, if not tens of thousands of years. Carolus Linnaeus was a naturalist in the 1700s. He created a naming scheme, which we still use today. It's called binomial nomenclature. It gives every organism what is referred to as a genus and then a species. He realized this was necessary because he began to notice that as he traveled, the same bird, for instance, might be called one thing in one location, but when he went to another location, the same bird had a different name. So he decided or derived to the fact that we need to make a change here. We need to know whether I'm in Germany or Austria or the Netherlands or Russia. When I'm talking about an animal, or a plant, I'm talking about the same thing. Modern biologists use what is referred to as the biological species concept to define what a species is. And remember, there are many, many different definitions of what a species is. But the biological species concept says that organisms that have the ability to interbreed and produce fertile offspring are a species. That can get problematic when we start looking at some of the some animals, such as the canines, for instance. Wolves and your family dog are considered different species, but they can breed and they can produce fertile offspring. So the biological species concept, which is the one we most readily accept in biology, still has some flaws or some issues that we have to deal with. New species form when some individuals can no longer interbreed with the rest of the group. Say, for instance, you have a group of squirrels that are running around happily all over the forest, and then an earthquake occurs, and the landmass splits, to where there's a 1 or 2 mile gap between this group of squirrels over here and this group of squirrels over here. And over the next 50,000, 70,000 years, this group over here is isolated from this group. And then those microevolution changes will happen within each population. And then, 75,000 years later, there's another massive earthquake. These two land masses are brought back together. These squirrels are now able to run in amongst each other, but they can't interbreed anymore. They have speciated. The group over here had some changes. The group over here had some different changes. And the culmination of all of those changes led to the fact that they were no longer able to interbreed. Therefore, we now have 2 species of squirrels. Asexual reproducing species and species represented by fossils never interbreed. Some organisms have the potential to interbreed, but don't do so in nature. We'll talk about that in a minute. Why, if 2 groups of, again, let's say 2 groups of mice don't interbreed, but they're running around, in the same area, why don't they interbreed? And that process where they don't interbreed is referred to as reproductive isolation. It is not always absolute, however, particularly in plants. We've already discussed the fact that plants do some really, I'm going to use the word strange things. When it comes to reproducing, they readily produce tetraploid or triploid organisms, where this plant will have, instead of the normal 2 sets of chromosomes, will have 3 or even 4. And that's one of the things we see in some of the ornamental plants we have. You cross a blue plant with a red plant and you get a purple plant because the blue and the red are equally being expressed. DNA analysis also helps us to define species. What we do is we take a known species, just a known species of rat, for instance, and we get a new rat that nobody's ever seen before, or is found in an area where it's never been recorded before. We take a blood sample from the rat, or we take a skin sample from the rat, we run the DNA, and we compare it to known rat species. I am a person who loves cryptozoology. I'm not necessarily a firm believer in cryptozoology, but I enjoy the subject. I like to read about it. I like to watch documentaries about it. And for those of you who don't know, cryptozoology is the study of unknown animal species. Many animals have been moved from what is considered cryptozoology to normal zoology. The mountain gorilla, for instance. For hundreds of years, there were rumors of gorillas running around in the mountains of Africa. But at the time, conventional biological theory said gorillas didn't live in the mountains. They only lived in the lowlands. So the mountain gorilla was a myth. It was relegated to cryptozoology until folks started start searching in Africa, and they started finding them. Same thing could be said with my favorite cryptozoological creature, Bigfoot. Does Bigfoot exist? I don't know. I want to live on a planet where Bigfoot exists. I don't know whether he does or not, but if he does, all the better for him. But Bigfoot at this point is a cryptozoological organism. If you watch those Bigfoot documentaries, every once in a while somebody will find some scat, which is what we call animal feces, or they'll find a hair sample, and the hair sample will be DNA tested, and the DNA tester says, well, I don't know what it is, but it's not a known species. And so then they say, but it's not Bigfoot. Well, how do you know it's not Bigfoot? You don't know what Bigfoot's DNA looks like because you've never tested it. But you've tested something here that doesn't look like human, it doesn't look like chimpanzee, it doesn't look like gorilla, yet you're saying it's an unknown primate. Okay, what could it be? I don't know. Again, I want to live on a planet that has a Bigfoot. But that's how we use DNA analysis. We compare it with other organisms, and if the DNA of two organisms is more than 97% identical, they are considered to be the same species. So that 97% is the threshold that we use right now. You take this animal and this animal, they may not look exactly the same. Maybe this animal is, 3 foot long and weighs 100 pounds, and this one is 8 foot long and weighs 700 pounds. But you do the DNA, and if they are 97% identical, then they are considered by science to be the same species. These reproductive barriers I talked about, why don't organisms reproduce? Why don't they interbreed? New species forms when a subgroup of a population can no longer breed and produce fertile offspring with the rest, aka my squirrels that were separated by the earthquake for tens of thousands of years. As that subject's gene pool shifts, it begins to follow its own independent evolutionary path. So you've got the group of squirrels over here that were making changes, the group of squirrels that were over here that were making changes, but not the same changes as here. And then 75,000 years later, they come back together, voila, new species. Reproductive barriers, we call these in science, we call them reproductive isolating mechanisms. It is a process that isolates groups from reproducing. So you have a list of these. They can occur either before or after the zygote is formed. These are going to be those before the zygote occurs. These are going to be the ones that are after. I want to look at these. Okay, yeah. It goes into the individual traits of each one on the next slide, so that's what I wanted to make sure. But again, barriers that maintain reproductive isolation between related species, these are things they're called pre-zygotic. In other words, before the sperm, before the sperm can implant or fertilize the egg. These are post-zygotic. You've got a sperm that has fertilized an egg, but then something happens here where we get what is called inviability. The organism either A, doesn't produce, it never forms a fully formed new organism, or if it does, that new organism is sterile and does not produce viable offspring. These are the 5 pre-zygotic mechanisms. Habitat isolation. They simply live in different environments, or they live in different places. The example used here is ladybugs feed on different plants. So you have one species of ladybug is feeding on this plant, another species of ladybug is on this plant, so they don't ever come in contact with each other. So habitat isolation. Temporal isolation. They're either active or fertile at different times. Field crickets. Different species of field crickets is the example used here, and they will mature at different rates. One species of field cricket is mature and ready to reproduce in the spring of the year, and another one is ready in the fall or in the midsummer. They're simply isolated temporally, as opposed to when are they active and when are they fertile and ready to reproduce. Behavioral isolation, they have certain courtship activities. We've all heard frogs croaking in the night. This time of the year, if you step outside and you live in the countryside, anywhere where there's some standing water, you're going to hear frogs croaking. A lot of things that people think are bugs buzzing and making noise are actually different types of frogs. One of the first groups of frogs that becomes active in the springtime is a group called the spring peepers. The first warm day, those peepers will come out from hibernation, and they'll be sitting on the edge of the water at night making these long peeping sounds. And it's the sounds that they are making the call that attracts the female. If the female is not interested in the call of a male, she's not going to respond to it. So frog mating calls are different. Whatever type of frog this is has a song. Whatever type of frog this is has a song. And only the females of this type will respond to that song. That is behavioral isolation. Mechanical isolation, either the mating organs or the pollinators are incompatible. The equipment doesn't work. It's what my zoology teacher at UALR said many years ago. I went to school in the same time frame as the O.J. Simpson trial. I know none of y'all remember the O.J. Simpson trial. Look it up, Google it. But there was a very famous saying in the O.J. Simpson trial where there was a bloody glove found at the crime scene. They tried to get O.J. to put the glove on. He showed that the glove wouldn't fit his hand. And his defense attorney, Johnny Cochran, very famously said, if the glove don't fit, you must acquit. Well, what my zoology professor said about mechanical isolation is, if the equipment don't fit, doesn't work. So the equipment's got to fit, mating organs or pollinators. Your textbook uses sage species having different pollinators, different types of flowers that pollinate different parts of the plant. So that is a mechanical isolation. The equipment just doesn't sync up. And then finally, you have gamete isolation because of differences in DNA, difference in other things. The gametes simply cannot unite. Again, mechanical isolation, maybe in gamete isolation, maybe the equipment does work. You know, the male equipment of a goat, for instance, is physically compatible with the female equipment of a sheep, but because of gamete isolation, you're never going to have a crossbreed between a sheep and a goat. They're too far separated. They have gamete isolation. The sperm of the goat will not fuse with the egg of the sheep. So these are the pre-zygotic reproductive isolating mechanisms. The post-zygotic isolating mechanisms, these are, yes, the equipment is compatible. Yes, they are fertile at the same time. Yes, they are in the same area. Yes, the sperm and the egg can fuse, but one thing is there is what is called hybrid inviability, which means the hybrid offspring fail to reach maturity. There are eucalyptus plants, eucalyptus trees in Australia that are of different species. Those species can cross-pollinate. They will produce seed or spores. Those spores then, however, will not produce new offspring. So yes, they've combined, but the product of that combination is inviable. It doesn't work. They never reach maturity. Hybrid infertility is where the hybrid offsprings are unable to reproduce. A lion and tiger cross is referred to as a liger. This is an infertile cat. You'll see here's the male lion, the female tiger. They crossbreed them. They produce this really huge cat. There was one of these that lived in the Bronx Zoo, I believe it was in New York. It was a liger named Tiny. I don't know whether he's still living or not. He'd be pretty old now if he is. But he was a cross between a 400 or so pound African lion male and a 450, 500 pound Siberian tiger. I think it was a Siberian tiger female. Produced Tiny the liger, and Tiny the liger weighed about 1,200 pounds. He was a huge cat. Supposedly was really timid, drank from a bottle, was like a great big house cat. Yeah, I'm not letting a 1,200 pound tiger loose in my house. And great, yeah, okay, yeah, he's still a wild animal. But again, he was not able to produce offspring because he was in fact infertile or sterile. The classic example of hybrid inviability is the mule. Human beings have used mules, for instance, for centuries, and the mule is a cross between a horse and a donkey. And when your mule begins to wear out and you need a new mule, you've got to go to a person who makes mules, who has somebody who has a horse, somebody who has a donkey. You crossbreed, you get a mule. And then finally, there's hybrid breakdown. That's where the second generation hybrids have reduced fitness. The offspring of the hybrid mosquitoes have abnormal genitalia, is in our example here. So we have the first generation hybrid between mosquito species A and mosquito species B, produces the hybrid mosquito. Those hybrid mosquitoes are then able to reproduce with either A or B or the hybrid group, either one. However, when they are born, all of their offspring of these mosquitoes have abnormal genitalia, and at that point, the group of organisms will cease to exist, is what I was trying desperately to say. So you'll get one generation of hybrids that are able to reproduce, but it stops after that one generation. Some spatial patterns. What is it that causes reproduction to cease? What is it that causes speciation? Excuse me, I've got to get something to drink. My throat's starting to get dry and I don't want to start coughing. So the first thing that can happen is you can have reproductive barriers. There are two different types of speciation that use reproductive barriers. The first one is called allopatric speciation, and that's where you have no contact between populations. This group over here gets isolated from this group over here, and over time you develop flowers that are all making nice little yellow flowers, and you have another plant that's making all little purple flowers. So that's allopatric. They are separated by something. Sympatric speciation, on the other hand, occurs when there is continuous contact between populations. See, all the flowers are over here, and you've got one group that's making purple flowers and one group that's making yellow flowers. But again, for some reason, the yellow ones tend to only pollinate with the yellow ones, the purple ones only with the purple ones, and that is sympatric speciation. The way my ecology professor talked about this many years ago was allopatric speciation happens in different places. Sympatric speciation takes place in the same place. So, allopatric speciation, what can separate populations? Rivers, mountains, glaciers, oceans. Anything that separates two populations, that prevents population A from getting over here to where population B is, would be an example of a geographical isolator. There's no gene transfer between populations, and allele frequencies become more and more distinct. Since they are only interbreeding with each other, again, it's very possible that in this area right here, before this mountain range was there, all of these flowers were able to interbreed, and you had purple ones over here and yellow ones over here, and vice versa. This mountain range then comes up, and for some reason, the purple became the dominant over here, the yellow became the dominant over here, and then finally, at some point, these things could no longer interbreed. Islands are wonderful places for speciation to occur. If you've ever studied any kind of evolutionary thought, or you've ever heard about it, you know about a man by the name of Charles Darwin. Charles Darwin is famous for his work on the Galapagos Islands off the coast of South America. Here are the islands. There are dozens of them. The larger ones have been named. Some of the smaller ones have never been given names. However, again, there's lots of animals lots of very unique animals on the Galapagos Islands. Darwin did most of his work with a group of birds called finches. As a matter of fact, on the island of Isabella, I think is where the Darwin Institute is. But there's a Darwin Institute of Study on one of these islands. It may be on Santa Cruz. But they've been studying these finches almost continuously for let's see, 18, 20, 120, about 140 years. The Darwin Institute, people have been studying these finches, and there are all kinds of finches. There are finches that are woodpeckers. There are finches that are bug eaters. There are finches that are seed eaters. There are all kinds of different finches, but they're all finches, and they all are the result of a island evolution. But probably the thing that the Galapagos Islands are most famous for are their tortoises. The tortoises of the Galapagos Islands. Many of these human beings have almost driven to extinction because, well, just to put it mildly, human beings are kind of stupid. Sailors used to go and land on these islands, and they would collect these tortoises, and they would bring them aboard ship. Sometimes they wouldn't even bring them aboard ship. They would simply turn the turtle over on its back, and the turtles are buoyant, and they would drag the turtles behind the ship, pull them up to the ship every once in a while, feed them to keep them alive, and then drag them behind the ship. That way they would always have a ready source of meat to eat. So again, human beings, kind of stupid. But the thing about these Galapagos Islands is there was at one time, and some of these are extinct, some of them are very nearly extinct, but on all of these bigger major islands, San Cristobal, Santa Cruz, Floriana, Isabela, Santiago, Fernandina, and Pinta, all of these islands had their own species of tortoise. You could tell if somebody brought one in and sat it down here, you could determine well, they have trouble sitting it down here. These things weigh 400 and 500 pounds. But you could tell where the tortoise came from just by its physical characteristics. And those physical characteristics are based on the topography of the island. You see this tortoise right here from Santa Cruz has this nice domed shell. There's no sort of hump in its carapace. That's what the shell is called. It's called a carapace. Right there behind its neck. It can lift itself up off the ground, but not all that far. But then if you compare it to the Hood Island tortoise, look at the difference in the shape of the shell. You've got this nice little sort of outtake. That allows that tortoise to take its head and raise its head very high. This tortoise over here can only get its head to about here because its neck's going to hit it. Its neck's going to hit its shell. But this tortoise can go from eating stuff off the ground to raising its head this high and eat things off the plants. And if you look, it has very long legs, and it can really lift itself off. And enough work has been done on these tortoises that we now know, and again, how did the tortoises get to the island? There's a phenomenon called rafting, where an animal will be near the coast, something happens, a piece of, they fall in the water, they're able to either swim to one of these islands, or they grab a hold of a limb or a piece of driftwood or something and make it from one island to the other. And again, the process is called rafting. And through fossil records, we now know that the island of Hispaniola, was called Hood Island in Darwin's day, was the first island to be colonized by the tortoises. We then know that the tortoises started moving. You see one group moved over here to Pinta Island, another group moved over to Santa Cruz, then the ones from the Santa Cruz Islands moved over here, and then these moved back over here, and then Santa Cruz Island also sent some representatives over here along with Santiago. The animals or the tortoises on Santa Cruz move over to Pizon, which then they moved back and forth. And then you have the groups of, on the island of Isabela. You see that the animals arrived here, but then they started bouncing down the island that way. Very interesting look at island speciation. And again, it's happening allopatrically, which means in different places, because these evolved separately from these, and they all have their own characteristics. Another way organisms can reproduce and be allopatrically speciated is isolated. They can be isolated. The pupfish of Devil's Hole Cave, which is right here on sort of on the border of California and Nevada. There's a nice little pupfish here, the Devil's Hole pupfish, the Ash Meadow pupfish. You see the Devil's Hole, which is again a cave system. Then the other ones here. It is again theorized that at one point all of this land was underwater, and that's where you found these pupfishes. And then as the waters began to recede and dry up and the desert formed, there was a little pocket of them left here and a little pocket of them left in Devil's Hole. And thereby, these are now genetically different from these. Still a very similar fish, but different species because they can no longer mate with fish from the nearby springs. The one in Devil's Hole is a unique species as opposed to the Ash Meadows species. Sympatric speciation occurs in a shared habitat. This is where populations diverge genetically, even while living together. Although, as this slide says, the habit may appear uniform, often there are many different microenvironments that can select for different phenotypes. Maybe in this general area right here, and we're just going to say, using this picture as a demo, that in this part, only purple flowers grow. Only purple flowers grow here, and only purple flowers grow here. Only the yellow here, only the yellow here, and only the yellow here. Maybe the soil here and here and here is more acidic than the soils here, here, and here. Maybe because of the mountain range and the way the mountain ranges is shaped, and maybe the sun moves in an arc this way, and maybe the mountains are a little lower here. There's a little bit of a hole here. There's a little bit of a hole here. Maybe more sunlight as the sun is setting. Maybe more sunlight is hitting on these flowers. So they're getting a few hours more sunlight. Maybe that's the reason you're getting these different plants. But again, microenvironments can play a role. Also, another example of microenvironment that can be sympatric speciation is very deep lakes. I'm not even going to try to pronounce the name of this lake, Ijaham. I'm not, again, okay, I said I wasn't going to try, and then I tried twice. But this is a lake in Cameroon, which is in West Africa. This is a very deep lake. It has very distinct ecological zones. It has areas next to the shore where the water is relatively shallow. Then the water gets very, very deep very, very quickly. But different species of fish are found in different areas of the lake. It just so happens that you're told that the larger fish feed near the shore, while smaller fish feed in deeper waters. The fish breed where they eat, they don't mix together, and therefore populations have started to acquire very recognizable genetic differences. Again, if you just look at the overall body shape of these fish, they are very similar. But then if you start to look at greater details, you see the deep water fish is different from the shallow water fish. Again, they're in the same lake, but they're separated by these micro environments of shallow water versus very deep water. Let's stop there for a few minutes. Let my throat rest, and I'll come back later and finish up this chapter.